Good morning, everyone. I'm Mohana Priya Jayavel. I work as a cytogenetic technologist at Northwestern Memorial Hospital. And this morning, I'll be presenting on integrated comprehensive genome profiling of meningiomas, a single institutional study. I have no disclosures. Meningiomas are tumors of the meninges, which are layers of tissue that cover and protect the brain and the spinal cord. They arise from the meningothelial arachnoid cells and are the most common primary intracranial tumors in adults, with the majority of cases being benign. However, some clinically aggressive forms of meningiomas are known to recur with significant morbidity and mortality depending on the location of the tumor. The common areas the tumor could arise include the parasagittal, olfactory groove, suprasellar, clivus, foramen magnum, and cerebellar. The best way to diagnose a meningioma is by an MRI or CT scan, and successful treatment options include surgical resection and/or radiotherapy. Here, I have an image of an MRI image of a meningioma arising from the arachnoid layer. The current WHO classification identifies 3 grades of meningiomas, which are grade 1, grade 2, and grade 3. The grade 1 meningiomas are benign and accounts for approximately 85 to 95% of the cases. There are 9 histological subtypes. They tend to grow slower and have distinct border. Their mitotic index is less than 4 mitosis per 10 high power field. The grade 2 meningiomas are atypical, accounts for 5 to 10% of the cases, and there are 3 histological subtypes. They must also meet 3 of the 5 minor criteria, which include sheeting, small cells, hypercellularity, necrosis, or prominent nucleoli. They grow faster and tend to recur and have a mitotic index of 4 to 19. The grade 3 meningioma accounts for approximately 1 to 5% of all cases. They are malignant, they tend to be aggressive, recur, and invade parts of the brain. Their mitotic index is greater than or equal to 20 mitosis per 10 high power field. And they also include molecular markers like the TERT promoter mutation or homozygous loss of CDKN2A or B. So in general, grading is solely based on histology for grade 1 and grade 2 meningiomas with an absence of molecular markers. In 2022, Driver et al. Proposed an alternative molecular grading system based on mitotic count, CDKN2A or B loss, and chromosomal copy number aberrations. Here, they list some of the common high-risk CNAs in meningiomas, which are loss of 1P, 3P, 4PQ, 6PQ, 10PQ, 14Q, 18PQ, 19PQ. So they assign 1 point for each of these high-risk CNAs and 1 point for CDKN2A or B loss. And in addition, they assign 1 point if the mitotic index is between 4 to 19, and if it is greater than 20, they assigned 2 points. Eventually, a total of 11 score points can be achieved, and depending on the score, the grades are assigned. So if the score is between 0 to 1, they are designated as grade 1 meningioma. If the score is between 2 to 3, they are designated as grade 2. And if the point is greater than or equal to 4, they are designated as grade 3 meningioma. So the purpose of this study was to evaluate meningioma cases at Northwestern Medicine from 2020 to 2023 and to review their demographic features, histopathology, tumor location, recurrence, genomic landscape, and clinical follow-up. A lot of large-scale genomic studies have identified recurrent copy number aberrations, gene fusions, and mutational profile of many brain tumor types. And in this study, we have utilized integrated comprehensive genome profiling using OncoScan microarray, next-generation sequencing, which targets 505 solid tumor genes, methylation array, and TERT promoter mutations to assess recurrent copy number aberration and to integrate histology grading and molecular grading to improve precision in meningioma risk stratification. So our cohort consists of 250 patients. A majority of them were diagnosed as grade 2. And based on OncoScan microarray, we identified 6 distinct cytogenetic subgroups of meningioma based on copy number alterations. The 6 groups are normal, single chromosome aberration, chromothripsis, hyperdiploidy, near diploidy, and hypodiploidy. So among these 6 categories, 4 of these categories harbored high-risk CNAs, which are chromothripsis, hypodiploid, near diploid, and hyperdiploidy. The chromothripsis was presented as an independent study earlier this year, and in this study, we focused on the remaining 3 categories, which are high-risk hypodiploid category, high-risk near diploid, and high-risk hyperdiploid category. Over the next few slides, we'll be reviewing a few aspects of our cohort. More than 50% of our cohort was female with a median age of 66, and in general, the incidence of meningiomas are known to be more in female rather than their male counterpart. It was most prevalent among the Caucasian population, and 50% of our diagnosed cases in this cohort are WHO grade 2. Among the high-risk groups, 51 cases belong to high-risk hypodiploidy, 53 cases were near diploid, and 20 cases were high-risk hyperdiploid, which includes triploidy and tetraploidy. Most of our cases had a mitotic index of 4 to 19, and we identified 5 anatomical tumor locations, which are cranial or spinal, skull base, sagittal skull base, sagittal non-skull base, and coronal. Moving on to clinical follow-up, our data at this point is limited, but however, 57% of our cases were primary, 40% showed recurrence, 88% underwent resection, and 33% underwent radiation. The genomic landscape of these 124 cases revealed a total of 1,145 CNAs identified using OncoScan microarray across all the 3 groups. The most common was loss of 1P followed by loss of 14Q, and to better understand the distribution of these CNAs, I have an overview of CNAs and mutation profile of 124 cases here. The CNAs are listed first on the left side, followed by NGS mutations. We'll be first looking at high-risk hypodiploid, followed by the near diploid category, and finally the hyperdiploid category. Over the next few slides, I'll be going through the data in detail. So our first category, the hypodiploid category, and based on Driver et al.'s grading system, we were able to reclassify 33 of our cases to new grades based on CNA scores. The most observed whole chromosome losses were losses of chromosome 22, X, 14, 10, 18, and Y. These losses are more frequently noted in high-grade tumors, and this category also had the most NGS data available, and the most commonly mutated was NF2, followed by ARID1A and TP53. Moving on to the near diploid category. Moving on to the near diploid category, 37 of our cases were reassigned to a new grade. The most common loss was 22, followed by 1P. We can see that this high-risk near diploid category, the CNA is distributed across all the grades, and copy number loss being more predominant than gains or CNLOH. Very few cases in this category had the NGS data available. And our final category, the hyperdiploid category, 13 cases were reassigned new scores. The common gains noted here were gains of chromosome 5, 12, 17, 18, 19, 20, and 21, which were predominantly noted in grade 1 and grade 2. And grade 3 is associated with loss or relative losses of chromosome 3, 6, 9, 10, 14, 22, which was predominant in grade 3. Moving on to mutation and methylation profiling, we had 17 cases that showed NF2 mutations, followed by ARID1A mutation, CDKN2A, MSH6, CDKN2B, and TP53. The right graph represents third promoter mutations and methylation. So 50% of our cases had third promoter data available, but less than 5% came back positive for third promoter mutation, and 66% of our cases matched methylation profile of a meningioma. Overall, we were able to reclassify our cases by integrating CNA scores and mitosis, which pushed majority of the cases to grade 3. 86% of grade 1 cases were moved to a higher grade, 59% of grade 2 cases were moved to high grade, and 28% of grade 3 cases were pushed down to a lower grade. In conclusion, meningiomas are characterized by 6 distinct cytogenetic subgroups, which are normal, single chromosome aberration, high-risk hypodiploidy, high-risk hyperdiploidy, high-risk near-diploidy, and high-risk chromothripsis, and are enriched by high-risk CNAs, even in low-grade tumors. Comprehensive genome profiling potentially regraded 66% of cases in this current cohort, and we suggest that CGP should be utilized clinically for molecular characterization and integrated in further WHO classifications for meningioma. Lastly, I would like to thank my mentor, Dr. Lu, who is also present in the crowd, and my whole team at Northwestern Memorial for their continued support. I would like to thank CGC for giving me this opportunity, and I appreciate all of your time.